Hi everybody, welcome to the SENS or SENS Research Foundation AMA, and we have a lot of people here already. Okay, that's great. So to kick off things, for people who don't know, perhaps you'd like to introduce yourself. Obviously the people from SENS Research, not everybody, we'll be here all night. Aubrey?
Hello, yeah, this is Aubrey de Grey. I guess most of you know who I am. I'm the Chief Science Officer of SENS Research Foundation, and I guess that my responsibility for the topic of today is that in the late 90s, I came across the problem of mitochondrial mutation accumulation and decided that the approach that had first been suggested in the mid-80s for addressing it was actually more promising than most people thought, and that people had essentially given up on it a bit too easily. So ever since then, I've been interested in pursuing it. We started actually funding work in that space in 2005. It took 11 years for us to get to the first meaningful breakthrough that was sufficiently significant to be publishable. But things have rather accelerated since then, so it's very timely to be having this campaign and this AMA.
Great. And Amutha, if you'd like to introduce yourself.
Hello, everybody. My name is Amutha, and I have been leading the MitoSENS program at SENS Research Foundation for the past couple of years. Previously, Oki, Matthew O'Connor, also in the AMA, he was leading the program. I joined SENS in 2013, like Aubrey mentioned, the major goal of the project is to test if allotopic expression of mtDNA genes that's expressing mtDNA genes from the nucleus can rescue pathogenic mutation with respect to function. So we have applied several strategies, and I want to tell that we've been successful at least with two of the genes that got published in 2016, the ATP8 and to a lesser extent the ATP6 gene. And we have we've just submitted a paper that widens the scope, and then we can show that we can achieve allotopic expression for all the 13 genes at some levels, some of them very good, some of them we will need much more engineering. And how can we take it to clinical trials would be to show a proof of concept in an animal model. And fortunately, there is a ATP8 mutant mouse available, and the goal of this project, like, you know, and the goal of this campaign, even the lifespan.io campaign, would be to validate this and you know, to create a transgenic mouse with the ATP8 gene in the nucleus and see how it improves function. That's our goal. So if we can take it to the animals, and our next step would be to help patients with mtDNA mutations.
Great. That's a great introduction. And we also have Oki, well, Matthew, but everybody calls him Oki. Over to you.
Hi, everyone. Matthew O'Connor, otherwise known as Oki, and I'm the Vice President of Research at SENS Foundation, and I've been working on this challenging mitochondrial gene therapy approach with Aubrey and Amutha for the past 9 years. Definitely been a huge chunk of my life. Yeah, Amutha gave a pretty thorough description of the state of the project. Really excited about the stuff that we've got coming out really soon, and looking forward to your questions.
There we go. That's it. Couldn't find the button. That's great. Thanks for that, Oki. And obviously, you probably don't know who I am. I'm Steve. I'm from Leaf. And we've also got Josh from Leaf. We're going to be doing the technical gubbins. And we also have Keith here, president. I believe he'd like to say a few words because he loves his own voice. So over to you, Keith.
Oh man,
Mr. President.
Well, yeah, everyone probably knows who I am, but Keith Comito, president of Leaf and Lifespan.io, which is currently running the crowdfunding campaign for the MitoMouse project, lifespan.io/MitoMouse. So I just kind of wanted to set the stage here. In addition to being like an AMA and where everyone can ask questions to the SENS team, which is going to be great. I was thinking it's also a little bit fun. We could do it a little bit like webathon style. So I'm keeping my eyes on that campaign. And if anyone donates while we're talking, we can shout it out. So for example, I see, Jordan Duncan just donated $25 to SENS for this campaign. So thank you, Jordan. Let's have a great conversation. Thank you, everybody.
Thank you, Mr. President. Good of you to join us. He loves it when I call him Mr. President. Oh dear. Okay, so for that should do it for introductions. So everyone should know who's who. Although for people who are new and may have joined us for the first time, perhaps someone would like to tell us a little bit about the SENS Research Foundation and the important work that's going on over there. Anybody want to take that one?
So I'm going to let Amutha do most of the talking since she's running the project. Go ahead.
Oh, so you want a general overview of SENS Research Foundation or the overview of the Mito project?
I guess maybe some overall context of basically, I think a cool way to maybe phrase this is how things have changed from maybe 12 years ago until now, in maybe using the MIT Technology Review as a good sort of then and now.
God, no, don't mention
circle.
them. Don't mention Jason.
Well, I mean, maybe sort of big picture SENS Foundation. It was founded 10 years ago by Aubrey and some other co-founders. It split off from Methuselah Foundation at that time to focus on actually trying to raise money to do the research. The vision being that we're funding research externally through grants that are geared towards our mission, which I'll describe briefly in a moment. And then also we built our own research center here in Mountain View, California, where we do a lot of our own research, including the entirety of the MitoSENS campaign, the MitoSENS project for the last 9 years. So in that regard, we've been successful at raising and spending in the $4 to $5 million a year range. We spend the overwhelming majority of that money on research, on funding research externally and intramurally, and our focus is, we're an aging research foundation, but our focus is on damage repair. So identifying types of damage that accumulate with age and finding ways to reverse that damage or remediate or otherwise make redundant, which is more the case for the MitoSENS project, the damage. So that in this case, the damage is the mitochondrial DNA mutations, and the repair is the engineered genes that we are putting into the nucleus to rescue those mutations. Is that a good overview for you guys?
That was pretty good. I mean, I've heard a few overviews in my time. That was pretty good. I'm going to say maybe a 7. What do you think, Keith?
Oh.
It's almost a holiday, so let's give them an 8. So
I'm British, so I'm innately cheeky. That's just how we are. So there you go. But yeah, that's a great introduction there. And right, let's have a look, because I'm just getting some questions coming in, which is always good. So we've looked at the basics.
I see some on Facebook coming in too, if you want me to mention any of those questions as well, Steve.
Yeah, one thing I did want to sort of ask all of you really, and that was how perceptions about aging research have changed in academia in the last decade. Has the acceptance of targeting aging to cure diseases become more popular in that time? What are your thoughts on that? I mean, I know Aubrey was somewhat vilified about a decade ago, but how are things in academia now? Is it sort of getting easier?
Yeah, all right. I guess I better answer this one. So, well, I mean, sure, it's massively easier. I think really we can say that the general approach of dividing aging into a manageable number of sub-problems and addressing each of those problems with a targeted damage repair strategy, that's become totally mainstream now. Of course, it's not the only approach that people are taking, but it's very much now accepted as a realistic way of going about things. I think that's become particularly true over the past couple of years, especially as we've seen so much benefits, such widespread benefits emerging from repairing just one type of damage, specifically senescent cells, eliminating those, which of course has been done now not only in the lab in mice, but also in early stage clinical trials by Unity Biotechnology. So yeah, I mean, the general concept, I think nobody's really trying to argue that it's unscientific or fanciful the way they thought a decade ago. It's very heartening.
Yes, I have a follow-up to that, Aubrey, that's related to some of the questions that we have. You know, obviously we have the hallmarks of aging that are, you know, categorizing, you know, aging into subtypes, like you just mentioned, you know, 7 to 9, depending on what you're looking at. Do you feel personally that we have to do, that there still needs to be a lot of foundational work to be done to further, you know, qualify the theoretical framework of aging? Or do you think we know enough right now that we should prioritize just doing what we can rather than doing that work, or both?
I feel pretty strongly that we know enough. In fact, I feel pretty strongly that we have known enough for at least a decade. And certainly the very fact that the classification that I first came up with more than 15 years ago now has stood the test of time so well is kind of evidence in favor of that way of thinking. You know, that's a long time to find nothing out, if you like. Yeah, of course, we may very well continue to identify new phenomena in terms of the accumulation of some or another type of molecular or cellular damage. But so far, all the ones that have been turning up over the past decade or so have been ones that fit very naturally into the classification that we already have. So if we look at the differences that exist between the different classifications that people have come up with, so for example, between SENS and the Hallmarks paper, these are not really differences in terms of what aspects of damage accumulate at all. In fact, if anything, the number of different categories that someone states is a poor guide to whether they're actually covering all the bases. So the Hallmarks paper didn't talk about cross-linking at all, for example. They just left it out by accident. But the point is that what the classification does is it constitutes essentially a guide to the types of repair strategy that can be developed. And so really the only reason why these different formulations sound as though they might actually be different from each other is because people are just choosing to give different amount of emphasis to different approaches to remediating the problem.
Yep, makes sense to me.
Right. So we've got a few questions. Just to let everybody know, which I forgot at the start. Duh, that's silly me. Just to let you know, at the bottom of the screen, you will see a chat bubble where you can type things in the chat, but also there's an option to raise hand. We're going to be taking some questions on the microphone shortly. So if you do have a question, feel free to raise your hand. You can also ask.
And before anyone asks any questions, let me just congratulate Jordan for being the first person to actually donate to the campaign during the AMA. He just announced the fact that he's done so. Thanks very much, Jordan.
Yeah, we're doing quite well. It's hopefully going to get some good research done. Okay, no problem. So let's take some questions if that's all right. Right, we've got a question from Chris Macaulay. He says, in which way do current mitochondrial enhancers like PDQ, MitoQ, or any of the other commercially available antioxidants differ in their effects to what you're working on? It's a fair question.
Maybe I can try with that. So all the antioxidants are only as let's say they remove the oxidative or relieve the oxidative stress in the organelle itself. What we are trying to do is it's a gene therapy approach. We are trying to place backup copies of the mitochondrial DNA because with time the oxidative stress or reactive oxygen species as a result of the oxidative phosphorylation complex causes or results in mutations of the mtDNA. And the mtDNA itself has much less repair mechanisms compared to the nuclear DNA, so it's not efficiently repaired, and these mutations keep accumulating with age. When you place a backup copy in the nuclear genome, you're essentially removing the site, like you're putting them in a safe location where you can generate the protein products that will still go into the organelle to perform its function. However, the genetic material for that is protected from the side products of the side products generated within the organelle itself. So MitoQ, all that is kind of transient. You would have to keep supplying these small molecules to prevent them, to help in any way. But if you place the genetic material itself away from the site of harmful product generation, then you're eliminating that factor. So that's the major difference, I would say.
It's a good answer. Hopefully that satisfied your question there, Chris. I see him in the call as well. But okay, if anybody's got a question they'd like to go on the microphone, please do raise your hand. Oh, we've got a couple of raised hands. Okay, let's have a look. Right. Okay, Nicholas. Now, there's a familiar name. If you'd like to go ahead.
Yes, can you hear me?
Yep, no problem.
Okay. It's the first time I'm using this app, so I would like to ask about, I don't know, it's not exactly about the MitoMouse project, but is about another project. I don't know if it's allowed to ask about another project, but it's about Matthew O'Connor presentation in Undoing Aging conference about Underdog company. The last time Aubrey came to Brazil 4 months ago, he told us that Underdog had raised like $2.5 million to start, and it needed $4 million. I would like to know if Underdog has raised the amount, like $4 million, how it's going, this process because it's a very important topic, and I would like to know what a regular person can do to help underdog to carry out its activities, and I don't know, raise money or funds, make some volunteer work for the company. I don't know how a regular person can help, because atherosclerosis, which is the focus of Underdog, if I correctly remember, is very important. My father has a problem with atherosclerosis, so I think like 1 billion people are waiting for something to reverse atherosclerosis. I know that MitoSENS and MitoMouse program can help too, but my question is about Underdog, how it's going, about that.
Yeah, sure. Yeah, I'll try to keep this brief so we can go back to mitochondria. But we have raised just about all of the $4 million that we need to launch, and we are in the very late stages of finalizing the corporate paperwork and dealing with the lawyers and the investors and such like that. So that should finalize in the next couple of weeks. So we've got commitments for all of the money that we need to launch. As you say, the company is a company in which we're developing drugs to target, and I want to bring the focus back to the kind of damage that we're targeting. The disease, the first disease that we're looking at is atherosclerosis, but the damage is oxidized cholesterol, namely 7-ketocholesterol, which Aubrey identified as a fundamental damage molecule. I first became aware of it when I was in graduate school, 17 or so years ago, and so I'd been working on this at SENS Foundation for part of my time for the last 4 years or so. So yes, this company is launching soon. In terms of being involved as an investor, unfortunately, we haven't gotten into the nascent ways of raising money from truly tiny investors. So right now, the minimum investment is $25,000. So it's more than what your typical person in a crowdfunding campaign can give. In terms of ways that other people can get involved, it's a good question, and we haven't yet created a mechanism by which volunteers can join. We will be interacting with the SRF education program. We'll be taking summer interns. Of course, that's only available to undergraduate students currently, but we may get involved in other volunteer programs related to that. We'll be launching websites and LinkedIn groups and such like that for it soon, so that people can stay up to date on what we're doing. So I think that addressed all your questions, Nicholas, and happy to take any more.
I just want to quickly jump in, say thanks for that, Oki, and also thank you to Martin O'Day and Mark Sackler, who donated while that conversation was happening. Martin listed as $30 donation on the page, and Mark $50 on, again, lifespan.io/MitoMouse. Thanks a lot. And I also kind of wanted to pivot on that question that was just asked to make it a little bit more broader to also SENS in general. I'm sure it also applies to LEAF as well. This is a question that I personally get a lot. I'm a regular person and I care about this topic. What is the most effective thing I can be doing? Like effective altruism as a regular person. So I'll just kind of open that to the floor. like SENS, Oki,
Okay.
Aubrey. Someone's passionate, but they don't have a lot of money. What can they do?
Well, okay. Yeah, if you don't have a lot of money, you can give a little bit of money. You can, I mean, seriously, every little bit helps on crowdfunding campaigns like this, donating to groups like SENS at SENS.org, like lifespan.io, participating in the conversations, getting involved in the groups. Right now we've got, I think, 2 different ways that you can tweet to raise money without having to spend a penny. There's 2 different hashtags. If somebody can remind me what they are, we can shout them out. That can raise, I believe, per tweet. One of them is $30 a tweet, and the other one is $1 per tweet. There's, in ways that you can post on a Facebook group to get $30 donations. There's lots of little things that you can do that, and on a day-to-day basis, participating, staying informed, reading our papers, reading the stuff that comes out on social media from SENS.org, on our different social media platforms. Lifespan is amazing on their social media side. So following, liking and sharing all of these things is important. And getting involved in volunteering. We have great volunteers who come down to the research center, and if you're not a scientist, we've got a guy who comes every week and helps clean up the lab and take out the trash and stuff like that, which is actually really helpful. People come to our symposiums and conferences and volunteer, doing little things. Every little thing like that helps. So lots of ways to get involved.
Great, that makes sense. I also want to shout out again, another donor just landed $50 from Balaz Suhajda. Sorry if I mispronounced that. So thank you for that, $50 to the lifespan.io/MitoMouse campaign. And to piggyback off of what Oki was saying, yeah, you can go to the SENS or the LEAF Twitter accounts, and you'll see that information. My own answer that I'd like to chime in is simply talking and keeping an eye on connections. That's something that everyone can do, and actually I find personally most interesting and most useful when someone says, hey, Keith, did you see this YouTube celebrity person was talking about aging? Maybe you should talk to them and you can make a video. Those things can have a massive impact, and that's something a normal person could just keep their eyes open and thread those connections together. So that's my answer for a low-hanging fruit that everyone can do. So I guess, Steve, back to you to field some questions.
I'm just going to add that I put in the Facebook chat the details for the tweet, the tweet there, and what you need to do. So if people want to help raise $30, it's a goodly amount for absolutely no outlay, just a tweet. Check the chat in Facebook, and that will show you how to do that. We've had quite a lot of questions in, and so people are obviously keen. Let's have another. We've got a lot of people from the audience. Okay, let's kick it over to Peter Grossman. Peter, go ahead.
Hi there, Steve. Thank you. My question is for Aubrey. And Aubrey, when it comes to the categorization that you've had of damage, the 7 categories, or as some have subsequently divided it into 9 categories, what do you think, which of those categories is the hardest obstacle to achieve longevity escape velocity, and how would you prioritize the other categories? Is it mitochondrial damage, or is it one of the other categories?
Okay, so first of all, Peter, it's great to see you again. It's been a while. Yeah, so let me answer that question in 2 ways. The first thing to remember about longevity escape velocity is that we get there with an incomplete, not 100% perfect panel of damage repair therapies. We just have to get enough postponement of age-related health conditions to give the scientific community the time to take the next step to stay one step ahead of the problem. And of course, that gets progressively easier over time, because the closer we get to perfection, the longer it takes for the damage that we still can't repair to get to a point that's pathogenic. So that's good news. Now, the reason I emphasize all that, just to make sure that everyone's on the same page here, is because one of the things we have learned from the recent progress against senescent cells is that there is a lot more crosstalk between the different types of damage than we might historically have suspected. I've always taken a rather conservative, pessimistic view on this, saying that basically we have to get pretty good at fixing all 7 of the types of damage in order to reach longevity escape velocity, because otherwise, each of these things can kill us pretty much on schedule on its own, however well we fix all the others. And that's certainly still true qualitatively. But the greater the magnitude of this crosstalk between the different categories, what that translates into is a greater level of imperfection that we can have in the panel in order to reach longevity escape velocity. So at this point, I don't think we could get away with just not fixing all of the types of damage at all. I don't think, for example, we could just reach longevity escape velocity without fixing cancer. That would just not happen. But I do think that the degree to which we need to be able to repair these therapies may actually be rather less than I would have historically expected. So that's nice. Nevertheless, since I think we do have to make some progress on all 7 of the categories, your question is still very well founded. And I would say at this point, the hierarchy of difficulty hasn't really changed from what I would have said 15 years ago. Namely, certainly the obviation of mitochondrial mutations is one of the very hardest ones. Of course, that's what the MitoMouse project, the MitoSENS project overall is all about. I still think we don't know how hard cancer is going to be. Back in the early 2000s, when I first started talking about SENS, we had basically no progress over the previous many decades in cancer immunotherapy. It was an area that was a real backwater, and people were just, seeming to be knocking their heads against a brick wall. And of course, a decade later, we had these enormous breakthroughs with checkpoint inhibitors and with CAR-T that are still we still don't know how far they can go, but they've certainly completely transformed that whole space. And we could end up in a position, 5 years from now, where cancer is, if not pretty much cured, at least is very much diminished as a problem, and we don't have to do any of these more elaborate approaches, like, for example, controlling telomere elongation that I've historically emphasized. So, that's somewhere where we don't know. The bad scenario, where we end up, finding limits to what cancer immunotherapy can do, and just not continuing to make progress there, that would leave us in a situation where the cancer problem remains a huge challenge. So that's definitely one. I think all of the others at this point are moving pretty well. Certainly the elimination of extracellular garbage, and also the replacement of cells with stem cells. These are areas that are moving forward rapidly. I mentioned senescence already, the removal of death-resistant cells. We've got half a dozen different approaches being pursued now in the private sector, and some of them already in clinical trials. The removal of waste products inside the cell has, of course, been a focus of ours for a long time, and we've already heard from a previous question about one of our favorite approaches to this, which is being pursued by our latest spin-out, Underdog. But we've also got progress in parallel with that in other areas, including the elimination of the garbage that causes macular degeneration using an enzymatic approach. That was a focus of ours for a long, long time, and we spun it out a few years ago. It's now about to enter clinical trials. And then finally, another very recent spin-out, Revel, which is working on the last of the SENS categories, stiffening of the extracellular matrix. So now, yes, I believe all of those are moving forward at a very healthy pace now. And so it really only leaves MitoSENS and OncoSENS as the big unknown. I'm definitely not saying that we've got to a point where SENS Research Foundation as a non-profit can actually just abandon all the others. For example, we have a stem cell project that we've only recently initiated, looking at using stem cells against Alzheimer's disease, which is quite an ambitious approach. There are various particular challenges in using stem cells in the brain that are, well, in the cortex anyway, that this project is trying to address. So yes, there's still, within each category, there's still a spectrum of difficulty as well. And that's, again, why we can't answer the question in a simple, elevator pitch answer.
Thank you, sir.
All right, our next question is going to come from James, who has had his hand up for a while. James, you
hello.
are good to go.
Hello, can you hear me?
Yes.
Wonderful. I'd first like to say what an honor it is to be speaking with Aubrey de Grey. I've been a fan for many, many years. And Keith's point about outreach is something that I think that this cause could do with a bit more of. So I will take it upon myself to recommend two products or people that have released products. One is called Eat Stop Eat by a man called Brad Pilon. Another is called Adonis Index by a man called John Barban. The reason I mentioned these two is because they are very much in on this cause, and actually it was them that told me about Aubrey in the first place. And I was wondering what you guys thought about the robust rejuvenation of a mouse as a demonstration of this idea, how far away we might be from that before private industry is able to take over this cause and it be a self-perpetuating cycle towards longevity, escape velocity. Finally, I would just like to say that I haven't donated on this particular chat, but I do donate to SENS through the UK subsidiary. I give £5 a week in order to give a regular donation to SENS of as much as I am comfortable affording, and that will not stop at any point because I've decided I could afford that for the future. So thank you for taking my question, and that's all I've got to say.
Well, first of all, thank you for the donation. That's really appreciated. And in fact, I don't know how many people there are on the call who are from the UK or from Europe, but I'm very happy to let everybody know that there is this mechanism for donating to SENS via our UK affiliate. If you are a European taxpayer on the mainland, then on continental Europe, then you can also do so because of a rather wonderful thing called, I think it's called something like Transnational Giving Europe, which is a kind of affiliation of charities from across Europe that allows tax-efficient donation across borders within Europe. So yes, anyone who wants to know more about that, there's a bit of information on the website, and you can also, of course, write to us for more specific information. To talk about just a bit about robust mouse rejuvenation then, yeah, I think we're pretty damn close now. I think it could be as little as 5 years before we get to a sufficiently comprehensive amount of rejuvenation in mice that we can really talk about having reached that goal. Remember that robust mouse rejuvenation is not the same as longevity escape velocity for mice. Reaching longevity escape velocity is actually more difficult for short-lived organisms than it is for long-lived ones, because basically we've got less time to make the next piece of progress. But robust mouse rejuvenation, as I defined it many years ago, which is essentially to add a year or two to the longevity of mice when the mice are already in middle age before you start doing anything to them. There, I think, yeah, we're getting damn close. And of course, really the practical definition of robust mouse rejuvenation is the point at which the publicly stated center of gravity of expert opinion is that, yeah, this whole rejuvenation thing is going to work, and it's only a matter of time before we get it to work in human beings. At the moment, we are still in a situation where out of all of the maybe dozen or so experts on the biology of aging who are willing to talk to the media at all and do a lot of public speaking and so on, I am still the only one who is able to tell it like it is by virtue of not being, fettered by the tyranny of peer review, and the risk of being labelled as having said things that got people's hopes up unduly, things like that. So yeah, I think that we're likely to get within the next 5 years to the point where it is safe for my more constrained colleagues to start talking a good deal more optimistically, and that is when things are really going to start moving. Of course, when you mentioned the private sector, that's already happening. We've got an explosion of investment interest in this area, which is why SENS Research Foundation has been able to spin out half a dozen of our projects so far into startup companies, and why literally dozens and dozens of other companies with very closely aligned technology have also been able to emerge and get well funded. This is already definitely happening.
Thank you, Aubrey. And could I just add that those affiliates in the UK and the EU, I don't think that they're clearly advertised as much as they could be. I think that is something that we could improve upon as a movement, and in order to make it easier for people to donate. But thank you very much for answering my question and for all the work that you do.
Well, thank you very much for that great question. And just to echo Aubrey, thanks for regularly supporting SENS Research, I should say, not to use the abbreviation. Okay. Well, we've got a few more questions. We've got some coming in from Facebook as well. So let's have a look. This is one about Foxo. So we've got a question from Tony, who I believe is also in the call with us. His question is, has SENS worked out how to improve FOXO4 to ensure cells don't go into chronic senescent cells or cancer cells? I think he means change into chronic senescent cells or cancer cells from the P53 gene.
All right, let me give a general answer to that kind of question, not just to that specific question. So no, we don't work in that kind of way. This is, perfectly legitimate and valid work that a lot of other people are pursuing, and that's one reason why we don't do it. We're always interested in focusing and prioritizing the things that are being neglected by other people. But the other reason is that that kind of approach is more along the lines of slowing aging down, helping the body to protect itself better, etc. Whereas our focus is always, as Oki mentioned at the beginning, on rejuvenation and actually repair damage that has already occurred. So this is not our focus, but that's not to say that we don't think it's valuable. It certainly is.
I can piggyback on some of the earlier questions that are also in our collected document of questions people wanted to ask. So you talked about the private sector. The flip side question is generally, how important is the policy side of this? Do you see, some people in the community have said, forget about the government, just blaze ahead. And there's other people in the community that says, no, we really need to bring governmental organizations on board. A very broad question here, but what are your opinions there?
Right, it's an extremely important question. I believe that that's the third wave of persuasion that we've got to start really focusing on around now. The first wave was to get the scientific community to understand what the hell this was all about, and that took a while, but it's definitely done now, as I mentioned earlier. And the second wave was to get the private sector interested, especially investors, so that it became possible for these academic projects to be funded for profit rather than just by philanthropy. And that, as I just mentioned, is very much in full swing now. The sector is exploding exponentially. So now I feel, yes, absolutely. Now is the time when we need to be getting policymakers, decision makers of all kinds to be much more focused on this. Now, I do not think we can really go route one and say, this is something that is a political area, an area where people need to go out and have policy commitments to, to spend money in this area. Because at the end of the day, that is all about, persuading politicians that there are votes in that. And to be honest, right now, it's not obvious that we can make a case that there are votes in it. Ultimately, politicians will not do anything unless it's going to help them get re-elected, or unless they think it is. So I don't think that's really the way to go. But that doesn't mean that we can't talk to these people. It certainly doesn't mean that we shouldn't talk to these people. It's just that the emphasis, I believe, that we need to have is more at the level of forward planning. The huge danger that I see is that when we get as far as robust mouse rejuvenation, never mind longevity escape velocity, we will see a really dramatic, incredibly sudden change in public expectation, public anticipation of the time frame for reaching actual longevity escape velocity, right? So in other words, that means, bearing in mind what I said a few minutes ago, that within the next few years, we can see a scenario in which, as a result of the change in what experts in the biology of aging say publicly, and consequently what opinion formers of other types, like, Oprah Winfrey, start saying, that we will see public expectations switching incredibly suddenly from a point where they expect to live, only a few years longer than their parents did, to a point where they expect to live essentially indefinitely. And that has enormous policy consequences, especially economic consequences. It changes what kind of life insurance people want, what kind of health insurance they want, how they're going to, arrange their inheritance arrangements, pensions, everything like that. And these are big ticket items. So the economy is going to have to be basically, redesigned from the bottom up overnight. And people had better have thought about how to do that before that happens, or else it's going to be unnecessarily turbulent, which would be a bad thing. We would save fewer lives than we otherwise might have been able to. So, of course, there are plenty of other reasons why politicians and policymakers need to start paying attention to all of this. Getting ready, for example, to front load the investment in infrastructure development and training of medical personnel and so on, so as to make sure that these therapies will be able to be delivered to everybody who's old enough to need them, irrespective of ability to pay, as soon as they actually are ready for prime time at all, because of the economic benefits that will arise from that. But to be honest, all of this has to be thought through by the entire community of people who have an influence on the direction of public policy. The private sector is all very well. There's plenty that the private sector can do in terms of being the leading edge of all of this, but it can't do it all.
Great answer, Aubrey. I largely agree for my part, and once again, bringing up the classic case study that I bring up a lot that everyone should check out. The history of the cancer advocates as detailed in the book, for example, The Emperor of All Maladies, and how they basically followed that same roadmap. Research first, building up grassroots support, and getting some private money, and then eventually that catalyzing a great societal public change. And I'm actually optimistic that we might be able to get some more buy-in on the policy side maybe a little bit earlier than it might seem, because of the adjacency to other issues that are currently kind of very vociferously discussed, like the future of healthcare, etc. Just as a tactical thing, I think it's much easier to convince someone that they're already on your side and just don't know it yet, rather than to try to get them to switch a position. I think there's opportunities there. But,
yeah, very well put. Very well put.
Yeah.
Let's have some more mitochondrial questions, because this is supposed to be what it's about. In fact, Keith, I think you asked one on the chat right at the beginning about double-strand breaks in nuclear DNA. Amutha, can you see it?
Yes. Yeah, so I see it right here. I can read it out if you'd like. So this was actually from someone who commented on the campaign page, and they had said, will we need to make double-stranded nuclear DNA breaks to insert mitochondrial genes? If so, how will we prevent nuclear DNA from ligating with other chromosomes? I think you're muted, Amutha. Yeah.
Oh, okay. Yeah, okay. Now it's okay? Can you hear me? Yeah.
Yes.
So I think they're asking if we will be making double-stranded breaks in the nuclear DNA to insert the mitochondrial DNA genes. No. For now, we are approaching two ways to this question. One is the random integration approach. So, this gene therapy is already in place for several other genes, like, we utilize the AAV vectors. All our nuclear DNA have AAV integrating sites already in place. So we would place the mitochondrial DNA in one such vector, and then it will randomly integrate in some location in the nuclear DNA. The other angle that we are approaching is a much more controlled way, where we will integrate the mtDNA genes in safe harbor locuses within the nuclear genome. In that, we remove many of the negative factors like gene silencing, or the copy number, avoid random integration and stuff by putting in locuses like H11 locus or H-ROSA 26 locus. And this is one angle that we are pursuing independently in the lab, and it is that's our next goal, is to express all the mtDNA genes from a specific location in the nucleus, and then see how they perform the function. So we will not be making double-stranded breaks in the nuclear DNA to insert these as of now. Yeah. Hello?
Yep. We have another question. If the MitoMouse proof of concept is successful in mice, what's going to be the next step for MitoSENS?
So this is just one gene. So this MitoMouse, like I said, it has a point mutation in the ATP8 gene. And so there are several pathogenic phenotypes, in the sense that they have anxiety, they don't perform well under stress, they have fertility issues, and of course, lots of behavioral issues. So like I said, this is not a very severe mutation in ATP8. It's a subtle mutation, but it will prove our fact that, when you accumulate mtDNA mutations with age, the performance of mitochondria kinds of slows down. And if we are able to overcome that by placing nuclear copies of the mtDNA gene, it gives us much more confidence. And the next goal would be to expand this to all the other genes. And we have an ND6 mutant mouse available, and eventually the goal would be, I would say, is to place all the 13 genes in the nuclear genome and test for function.
If I can add a little bit to that, I would say that we're not currently working on a clinical product, but we might not be that ridiculously far away from taking a step like that for mitochondrial disease. Like Amutha said, we have stuff that works great in a small number of the genes, and we know that our technology is already fundamentally superior to what is in the clinic today. And actually, Amutha and I and some other people have been talking over the past year and even just yesterday about strategies. What's the best strategy? When should we, aggressively start, pushing to try to develop gene therapy products that we can take to the clinic at a small scale to start helping people who are suffering from mitochondrial disease. And then as we continue to improve the technology, expand that to all 13 genes, which would be able to help all of us with aging. So that's our general strategy of how we think that this will roll out, is it'll be one gene at a time for people with individual congenital mutations in mitochondrial disease, and then spread from there. And we're actively strategizing on how we can start breaking into the clinic with with our technology now and in the near future.
And speaking of the clinic and translation to people, which is obviously ultimately where we're going, I'm going to ask a question which does come up quite often. How would MitoSENS get through the FDA, given that aging is not currently classified as a disease? Is this a huge barrier to progress, as some suggest?
Well, I think, okay.
No, go on, Amutha.
I just wanted to say that it's not any different from other gene therapies, but then there are added layers of complexity that you have to engineer the mtDNA genes so that they become functional from the nuclear DNA. Otherwise, it's very similar to many other gene therapies, except, yeah, you're talking of placing 13 genes in the nuclear genome. And that is a hurdle, yes. Also, they're going to be transmitted down the line. Like, if you make germline mutations, then these are going to be transmitted down to progeny, and then they would, in fact, have the protective factor of having a set of mtDNA genes in the nucleus, and that's how I see it. But there may be hurdles around that.
So to cover a couple of other aspects of your question, so yes, the things that Amutha mentioned are very true, and they pose additional hurdles or greater hurdles relative to standard gene therapy applications in the clinic, especially the fact, I think, that we're putting multiple genes, we're proposing to put multiple genes in at the same time. The germline question is one that, of course, applies to gene therapy in general, and the approaches to addressing it are the same, I believe. So, for example, either ensuring that the genes simply did not insert into germline cells like sperm stem cells, for example, or alternatively, that they could be excised by application of some drug-inducible genetic manipulation, for example. However, the other aspects of this therapy that I think you're getting at are also worth mentioning. First of all, the idea that aging is not a disease, and therefore that this would have difficulty getting through FDA approval. You have to remember that, just like so many other aspects of SENS, this same technology is absolutely applicable to early onset things that are very much called diseases. In particular, there are some mitochondrial mutant diseases that affect kids and young people, and that can only be addressed by introducing all 13 protein coding genes as backup copies in the way that we're doing, just in the same way as the mutations that accumulate during aging. So there we kind of sidestep that problem. Another thing to mention, which is not specific to MitoSENS, but is worth mentioning in case anybody doesn't know about it, is that actually the whole idea that aging is something that the FDA doesn't approve drugs for is more or less history now. That's been more or less fixed, largely due to the supreme efforts of Nir Barzilai and his colleagues, who were able to formulate a rather long-winded and complicated, but nevertheless effective description of aging in all but name that have allowed the FDA to define clinical trial endpoints in these terms. The clinical trial for which this was done is one using metformin against aging, which has very recently, in the past month or so, become fully funded, so that's going to happen. But yes, as I say, this only came into existence as a result of extremely painstaking discussions between the scientists leading this thing on one hand, and on the other hand, the FDA themselves. So I believe that now we're in the very much improved position where that same definition of clinical endpoint, that is aging in all but name, can be just copied and pasted into any other clinical trial for any other treatment that anyone might develop. this, of course, has huge implications in terms of the incentive structure in big pharma for actually investing the time and effort and money to develop such things.
All right, we have another question. Could the mtDNA copies placed in the nucleus cause conflicts with gene expression, causing the histone to turn off?
Oh, yeah. So that's why it's important to know where the mtDNA is going. As I mentioned earlier, we can test the conventional AAV vectors that have been used for other gene therapies, which we know that, you know, it works well for other gene therapies, or we can place them in specific locations within the nuclear DNA so that it doesn't impede functions of other genes in the nuclear genome. The one other thing we are also testing in this MitoMouse is that angle also, that when you place it's actually a foreign gene for the nucleus. So what is the immune response of placing such a gene? Those studies are in the MitoMouse project as well. We will analyze how the animal responds to expressing these foreign proteins.
Makes sense. I'm just going to jump in for a second. I see that we've had a big influx of people on the stream. So I just wanted to contextualize for those of you who don't know what's going on. We are discussing, amongst other things, the MitoSENS project of the SENS Research Foundation to essentially back up mitochondrial genes in the cell nucleus and thereby help prevent mitochondriopathies that can impact aging, and in support of the lifespan.io/MitoMouse campaign. So you can go there if you want to help, and we'll read your name out. I also see another question that popped up in Facebook, and this is basically the new CRISPR improvements that were just announced a few days ago, you know, the prime editing. How can you see this, if at all, assisting in MitoSENS or the SENS agenda, you know, thoughts on how this can help the process?
Yeah, so we just, for this reason, we journal clubbed the paper just yesterday, but I have to tell that CRISPR hasn't been effective in editing the mtDNA itself, just because of the repair you need the repair mechanisms in place when you introduce CRISPR-Cas9 to make the changes that you want. The other hurdle is also, people have tried it. There is one report which, it hasn't been validated. But introducing the big Cas9 protein into the mitochondrial matrix has also been difficult. So in that sense, editing the mtDNA itself is far away, I would say, unless we come up with better technologies. But the paper that came out is very detailed, and it can create all the protein transversions that happen in the nuclear genome. And in that sense, it's a nice breakthrough.
Yeah, I just want to add to that a little bit. So some of you out there may be under the impression that there is basically no DNA repair in mitochondrial DNA, because that was actually the dogma for quite a while. It was discovered back in the 1970s that one very fundamental type of DNA repair is indeed missing in the mitochondria. But then after that, it was discovered that other types of DNA repair are actually present. So, it's a complicated story. And that's actually something that may end up making this prime editing story quite interesting, because the type of DNA repair that is involved in, that it has to be recruited in order for the prime editing thing to work, is actually not the same as what we would typically expect in standard CRISPR. So that's one thing that may actually end up making this worth looking at. Perhaps not by us, though. This is something that other mitochondriologists are also going to be quite interested in.
Fascinating stuff. We've got a raised hand, and Alexandra, would you like to go ahead?
Yes, can you hear me?
Yeah, yeah.
My question would be about kind of basic working places. You might hear about basic income. It's a popular question now um, on how to allow people to do some volunteer work and to improvement in social area, um, having a basic income and in free time to improve uh, some on commercial area. So that's my question. Does anybody hear anything about basic working places which will take less time than usual job and will allow to invest um, a labor our own workforce to the project. Thank you.
It was a little bit difficult to hear. Steve, did you get all that? Can you maybe
Yeah, I didn't understand the question at all.
Sorry, yeah.
I heard some stuff about the universal basic income, and I think about ways to volunteer to help support this.
Yeah, I guess if that's correct, a way to phrase this that could promote discussion is, how do we see what we're trying to do as a field implicating income inequality and that sort of thing?
Well, I think part of the idea behind experimental ideas like EBI was around what Aubrey was saying earlier of, as we hopefully move into something like an ageless society, and with drastically improving lifespans and reductions in disease and improvements in quality of life, how do we change the social structures and the work structures, how is all of that going to be managed? And so I don't know that we're in a position to state like, okay, this is the fix for the pensions, or this is the fix for for poverty or anything like that. But like Aubrey said, it's not too early to be experimenting and speculating, and that policymakers should be considering interesting ideas like EBI. And let's see, I'm looking at the chat. So as people are talking, I don't know, this is straying a little bit away from aging, I think, when you're talking about not having enough work to do in society, perhaps because of too much automation and such like that. But I think experimenting with social programs and ideas like this is already happening, and probably isn't happening fast enough.
I see. I also see Alexandra was clarifying her question a little bit. And I think a follow-up to this might be one aspect that maybe we don't talk about as much as we should, since we have our head in the research and the biology, is the quote-unquote social determinants of health. So do we have any thoughts maybe that we can discuss on, might it be better for the overall health of everyone and the rate of aging if people, had shorter work days, or ways to mitigate stress in their life and that sort of thing? So I think that was more towards the spirit of Alexandra's original question. Basically, your thoughts on the social determinants of health and how much people should prioritize and alter their lifestyle based on those sort of findings, maybe would be a good question.
Yeah, I think there's good evidence that that having strong social networks is good for health. Having strong social networks and not being ridiculously stressed out. I don't know if we're the best people to preach at that, because we're always working around the clock here to to advance the work that needs to get done. And Aubrey's always on a plane, so I don't even know if he has circadian rhythms anymore. But probably everyone out there should be resting and exercising and eating well and not overworking themselves. But we all are.
Well,
So
it's well known that scientists run on coffee anyway, Oki. So, you know, just keep it coming, right? Sorry,
I see a question on the chat.
Yeah,
I see
yeah.
you talk about regulation and expression of the.
Yes, I see that from Anais. On top of moving the gene inside the nucleus, how do you control the regulation and expression of this gene? Will the expression level of the gene stay the same? Do you silence the original gene in mitochondrial DNA? No, for now we don't silence the genes in the mtDNA, but that is possible. It is there are experimental conditions that you can entirely eliminate mtDNA. Once you have all the 13 proteins working, we could show this at least in vitro cultures in what we call row zero cell lines. But for the regulation of gene expression, for now we are using viral promoters. We are also working on several endogenous promoters. The promoters used by rest of the 1400 genes that are encoded by the nuclear DNA, and we also have ways of regulating the expression. It is possible by just changing several factors, the promoters, and upstream 5' or 3' UTRs. It is possible to regulate the level of expression. Yes.
That makes sense. Amutha, can you maybe speak a little bit more about was there a reason or why you started with these 2 of the 13? Is there sort of a difficulty order of the remaining 11? You know, that sort of stuff.
Yes. Yes. So some of the general features of all these 13 genes is that they are multipass membrane proteins, and each of the protein has different numbers of transmembrane domains, let's say. So ATP8 turns out to be the smallest protein in all the 13 proteins, and it has only 1 transmembrane domain. So we think that's why this was the first one to achieve efficient expression. But having said that, I can also say that we have several other genes, like I pointed out earlier, that we have positive data for a handful of other genes, whereas some of the bigger genes, such as cytochrome B or COX-1, is going to need further engineering. Their hydrophobicity value is much higher, and they are also much bigger proteins. So we will need further engineering for them. So like I said, so ATP8 is the smallest protein, and it has 1 transmembrane domain. So it's only reasonable that it worked out first.
That makes sense. Thank you.
Okay, I see we have a raised hand. James, if you'd like to go ahead.
Yeah.
Oh, sorry there, James. Go ahead.
Hello, can you hear me again?
Yeah, go ahead.
Yep.
Fantastic. In his TED talk some years ago, Michael Ray, the co-author of Ending Aging, suggested that there was essentially nothing we can do to prolong our lives at the moment other than things that would shorten life. So maintaining a low body fat percentage, not lying on train tracks, not smoking, that kind of thing, would all be the things we could do now in order to make sure we don't die early. Is that still the case? Or has science advanced to the point where there is something that can be done to increase that? And also, on the topic of mitochondria, with maintaining mitochondrial health, is there any impact on the macronutrient ratios that we have, so the amount of sugars compared to fats and proteins, that may or may not increase or decrease longevity of the mitochondria specifically?
So I can talk to the mitochondria part. So it's well established that mild exercise for 20 minutes a day will increase the number of mitochondria in your body. So that is known now. For the macronutrient, yes, maintain a healthy diet, no high carb, high fat diet will help, I think. But I think Aubrey can address to the rest of the question on the larger scheme of things, how things can work.
Yeah, I guess so. It's still pretty much true that there's nothing really we can do. I mean, of course, I always have to make sure that people understand what that means. We certainly do know that there are plenty of easy ways to shorten one's life expectancy, smoking or getting seriously overweight or having a very, restricted diet that is short of micronutrients and so on. But, over and above basically living the way your mother told you to, we are still in the situation where there's not a lot we can do. We certainly do have reasonable evidence to the effect that we can somewhat protect ourselves against an imperfect lifestyle by certain things that are essentially mimics of calorie restriction. So I mentioned the TAME trial already. Metformin has been reported to extend life, in other words, postpone all-cause mortality, even though, of course, it has predominantly been given to people who have a handicap, namely they have diabetes. So this is quite exciting, and a lot of people think that the main mechanism of action for this is that metformin impedes the activity of the respiratory chain and thereby mimics calorie restriction in some ways. So yeah, this is the kind of thing that hints at the possibility that we might be able to do something. However, we always have to remember that calorie restriction itself almost certainly will only extend longevity in long-lived species like humans by a very small degree, if at all. So we're talking about health span rather than lifespan, and we're probably talking about a pretty modest impact anyway. Nevertheless, let's not diss it. This is better than nothing. But clearly the emphasis has to be on hastening the development of therapies that can do a hell of a lot better than that.
All right. We have another question. What would your response be to David Sinclair's assertion that epigenetic changes are upstream of pretty much all the hallmarks of aging and therefore the SENS approach?
Yeah, so I saw that. So I haven't actually read David's book yet. David and I are great friends, and he really ought to have sent me his book some time ago, but he never quite got around to it. And then I got given a copy at RADFest a couple of weeks ago, but I haven't had a chance to read it yet. However, yes, I know that David is quite keen on the idea. I think he calls it the information theory of aging or something like that. That epigenetic noise, the accumulation of damage largely generated by, as a side effect of DNA repair, may be significant. At this point, I'm still skeptical that that's going to be a significant player. Some years ago, we funded a very important study at Albert Einstein College of Medicine, looking at the rate at which epigenetic noise actually accumulates, which is a very, very hard thing to look at in long-lived species, especially in multicellular species in general. But we made some enormously good progress, technical progress, in terms of being able to actually study the amount of epigenetic noise in tissues by looking at the epigenetic sequence of individual cells. This took a huge, I think, 3 orders of magnitude improvement in the sensitivity of the techniques. That was the first step, but we never got far enough to really get a definitive answer to the question, does epigenetic noise actually increase with age throughout adulthood at a rate that could be significant? The tentative information that we got was that it probably doesn't, but it was very tentative. So I'm definitely all for more work happening in this area, and to the extent that David's comments are going to encourage that to occur, I'm very happy about that. However, as I say, I haven't read the book closely yet, so I shouldn't probably comment any further because I'll be overstretching from what I actually know.
It's a fair answer, isn't it, folks? It's a fair answer. The whole cause-consequence debate still continues. I'm sure we'll get to the bottom of it sooner or later. We've got another question from Matt Johnson about mitochondria. He says, how plausible would it be designing mitochondrial genomes from the ground up to remove faults or add benefits? I think it's only 32 genes, 16,000 base pairs. So could it be a low-hanging fruit for synthetic biology as it progresses? It's a good question.
Yeah. Well, let's say we've brainstormed about this, and yes, you could replenish mtDNA or wild-type mtDNA into the mitochondria. So there are lots of techniques now in place. There's even organelle replacement. It's called MRT, or mitochondria replacement therapy. It's been employed predominantly in the fertility space. But if we get to a point where we can either let's say transplant organelles, mitochondria, healthy mitochondria that has been lab generated, or if we can send in wild copies of mtDNA to tip the heteroplasmy or the number of wild type mtDNA, there is a possibility. So there is work going on in this area. They can send in small plasmids as of now, reporter plasmids with GFP in it, but people haven't tried the entire mtDNA as of yet.
All right, well, that's very illuminating. What are your thoughts on inducing mitophagy as a strategy to weed out mutant mitos? Some people try to do that with supplements.
Yes. So this is also another great approach, that if you can find small molecule modulators to specifically recognize, let's say, just dysfunctional mitochondria. However, the pathways that you would be targeting, they may crosstalk, and we would have to find the right type of molecules to target only diseased mitochondria and not everything indiscriminately. There is some studies going on in this area too. People are trying to find PKA inhibitors or in the how to modulate ATP-IF1 gene that they call. So yes, it is going on in this area.
Yeah, just to amplify a little bit on what Amutha said. So as some of you probably know, my very first paper in the biology of aging back in 1997 was the first time suggesting that the clonal expansion of mitochondrial mutations might actually be driven by kind of maladaptive autophagy. In other words, the preferential destruction of wild-type mitochondria and the preferential protection and essential survival of mutant ones. This is a hypothesis which still, I think, remains unproven, but also consistent with all the data. And one thing that we've certainly been interested in the possibility of is somehow biasing the selective balance in favor of wild type rather than in favor of mutant, so as to eliminate mutant mitochondria by manipulating autophagy of mitochondria. The evidence that exists right now suggests that this ought to be possible, and this protein IF1 that Amutha mentioned is a target that might be able to mediate exactly that. But at the moment, there's a hell of a lot that's not known about whether this is practical. It's certainly something that we would like to explore more.
Great, thanks for that. We've got another question here in chat. Are there some known reasons why the remaining mitochondrial DNA genes would have stayed outside the nucleus during evolution? Why would they have done that?
Please repeat that.
The question was, are there any known reasons why the remaining mitochondrial genes have stayed outside the nucleus during the course of evolution?
Yeah, so there are several theories people have propositioned towards this. One is the local need of high demand of these proteins that need to be produced for OXPHOS function. But remember that almost over 80 proteins of the OXPHOS proteins come from the nucleus. So it doesn't add up. The other angle is the extreme hydrophobicity of these genes. So you can find different pieces of mtDNA all along the nuclear genome. So it happens randomly throughout evolution. But to get a functional gene to be formed in the nuclear DNA would require acquiring all the correct promoter and the sequence correction for nuclear cytosolic translation as well. So I would say at this point but the thing is, it's probably the hydrophobicity factor and the complexity of the proteins itself that has prevented. Or it could be just an evolutionary thing. They are slowly transferring, and at some point it will transfer. That could also be a reason.
Yeah, for anyone who wants more information on this, I wrote a paper probably in, I think, 2005 in Bioessays, which is still reasonably up to date. There's only been a couple of important things that have happened since then. And this paper was a very comprehensive survey of all of the reasons that people had given, the examples that people had looked at that might suggest that it wasn't just hydrophobicity, and basically showing that none of those examples stood up to close scrutiny, and so that, you know, hydrophobicity is basically what it's all about. The consequence of this, of course, is that we have to find ways to address the hydrophobicity problem, and a lot of the things that we've been doing over the past decade here at SENS to get this whole thing to work have been along those lines, especially really our project essentially sprang into life back in 2006 or so, when a group in Paris figured out a way to largely sidestep the hydrophobicity problem. It wasn't by any means a complete solution, but it definitely helped. And most of what we've been figuring out ever since then has been ways to continue tweaking the system so that really viciously hydrophobic proteins can still be imported.
Mm-hmm.
Great. So I think we're kind of coming up on time now. So I wanted to remind the audience one last time that if you want to support this incredibly important work, go to lifespan.io/MitoMouse, MitoMouse, to help and donate. And I think one last question that I usually like to ask in these scenarios is very vague, which is, what question do you wish that we or the audience had asked you? And or what is it that you really want to leave people with about this work or the MitoSENS campaign that we haven't talked about?
Yeah, let me go for that one. So, I mean, I think it's with a project like this that's so challenging and so long term, it's important for people to understand the progress that's being made and to see that, you know, we are actually making progress. Most people don't work on these really challenging problems because they have to publish all the time in order to get their next grant application funded and to get promoted and get their next jobs and so on. And a large part of the reason why we set up the foundation as an independent charity that is funded entirely by philanthropy was precisely so as to escape from that tyranny of publish or perish. But of course, the downside is people need to see that progress is being made. And, it was a very long time. It was more than 10 years from the inception of the project before we were even able to publish our first paper. So I think one thing I ought to mention is that we do have another follow-up paper in review right now, which reports on some really big advances compared to what we published 3 years ago. Amutha's been quite downbeat about how little progress has been made, but I want to emphasize that that's because she's the researcher and she's paid to be, not to overpromise and underdeliver. We have made enormous progress. We're very happy about it. And the fact that we have this particular project coming up, moving for the first time into actual mouse rather than just working in cell culture, that is really another demonstration of the fact that we are moving really fast now. And, there's reason to be very optimistic about the MitoSENS project. We believe that once we have shown even initial benefits of one gene in mice, that the sky's the limit. We'll be able to move much more quickly going forward. So this project, even though it's relatively small as a proportion of the total budget of MitoSENS, is actually a really, really important step forward that we are delighted to be working with Leaf and Lifespan.io to make happen.
Great, that's exciting, and I'd just like to piggyback on what you said by letting the audience know. Again, we can't say it enough. 100,000 people plus die every day of aging, and if we can move the needle up by even 1 second, we'll save someone from horrible diseases. So don't think that your contributions do not matter. They matter immeasurably. Oki, Amutha, do you have anything you want to add on to that?
Yes. I just want to take a moment and thank Steve, Keith, for all you've done. I want to thank the Lifespan.io, Longevity Foundation, the Foster Foundation, all of you for supporting us, and all the individual donations. Everything, even every cent matters to us. So a heartfelt gratitude and thank you to you all. Yes, I can't thank you enough.
Excellent. Oki?
Okay.
Thanks for everybody for donating. We had some great donations during the campaign today. Thanks, last night when I went to bed, I think it still was under $60,000, and we crested that in the last 12 hours. So I really appreciate everyone's support, and I am glad that we get so many questions frequently in this webinar about how people can contribute to our mission, and I think we had some great discussion on that. To piggyback on what Aubrey said, do learn about and ask about the technology, the new stuff that's coming out. Try to understand how it's working, and read our paper when it's finally published. We've been working on getting this last paper published for the better part of the last year. So hopefully that saga will be done soon, and we can make it completely public in the very near future. So stay tuned.
Excellent. And of course, I think I can speak for everyone by saying, thank you, Oki, Amutha, Aubrey, of course, for, doing, in my opinion, the most important work in the world, and blazing the trail here. And I'm sure, you have the gratitude of millions of people, whether you see them on a daily basis or not. But I think that's, again, wonderful, amazing work. Steve, anything else you want to say before we wrap up? Or?
Sure, yeah. Just want to, a word of caution to everybody out there watching. We are approaching Halloween, and aging is a horror story, but we are being stalked by the Mitochondria. So keep an eye out for that. Yes, that's one of Oki's creations, I'm afraid. I wonder, does that make him Dr. Frankenstein? Perhaps. We don't know. But I just can't behave myself. Thanks for everybody who's joined us this evening anyway. It's been fun as always, and thanks to obviously SENS Research for taking the time out of their day. yes, it's late here in the UK, and we're all thinking about having a nice beer afterwards, and it's still in the morning over in California where they are. Sorry, guys, you've got to work the rest of the day. But
Thank you.
thanks for everybody for joining us, and do check out lifespan.io/MitoMouse, which is the short and cool way of getting there. We do that a lot, and hopefully we'll get there. We're very close to the stretch goal now, so every little helps. And do check that out. And as I mentioned earlier, there are ways of helping us raise money for that by using Twitter. There's more details on the campaign page how you can actually donate $30 without actually donating anything. So do check that out too. So thanks very much, and hopefully if you're having a Halloween party today, as some people seem to be doing that, it's a UK thing maybe, I don't know, but we always pick the nearest weekend so we can get drunk. I don't know what you do in the States, but there you go. We're practical people over here in England, so and so there you go. And I'm off to go and have a party now, and I hope you all have a good and healthy, productive day, and we will see you again soon. Thank you very much.
Thanks, everyone.
Thank you.